Hello and welcome to a quick overview of some things related to meiosis, which some of you are starting to learn about by drawing some of the steps and taking a look at some sources to consider some questions. This video is meant to be supplemental to your own work and research to help you make sure that you understand some of the new terms and ideas going on in cells and genetic variation. So let's start. So in a nutshell, we know that there are cells, and cells have an organelle called the nucleus, where we find chromosomes. Now, we sort of simplified the structure of a chromosome during mitosis, and what we said was that there was 1 and 2 copies of each chromosome that was duplicated during the S phase of the cell cycle. And during mitosis, this is taking a little bit longer.
Please excuse the interruption. Stowe High School boys ice hockey is now dismissed.
Excellent. So, boys ice hockey is dismissed, but also during mitosis, kinetochores attach to the sister chromatids, the 1-2 copies of one chromosome, and they separate them, which results in 2 cells after cytokinesis and telophase having identical copies of chromosomes. So each of these is identical. But we now need to consider how we get chromosomes in the first place and how that's related to genetic variability. So within cells that we call gametes, sperm and egg, there is a nucleus and there are chromosomes. However, during meiosis 1, we have chromosomes lining up on the metaphase plate that are arranged by a thing called homologous chromosomes. Sometimes we refer to these as homologs. And what we need to know about homologs is that they're a pair of chromosomes that are purely 50% paternal. I'm going to draw blue here, and maternal. So 50% of a homolog is maternal, and 50% is maternal. Now, we should note that there are 2 copies of each chromosome on either side of the metaphase plate. So right here I have 2 copies of a chromosome that's paternal and 2 copies of a chromosome that is maternal. And because chromosomes line up on the metaphase plate in pairs, sometimes we have random assortment of different homologs. So this is one of the ways that genetic variability can be introduced in gametes, because you can mix and match or rearrange maternal and paternal chromosomes. If you need to know, we have 23 pairs of these homologous chromosomes, 22 of which are maternal and paternal, and then the last pair is either XY, most of the time for biological males, or XX for females, but that are biological. During meiosis 2, after we separate these homologous chromosomes from each other, we then have cells that are haploid. And haploid means that they only contain half of the genetic material of the parent cell. So we still have, though, duplicated copies of those chromosomes. This is sort of a jump. So we started off with a cell that had 4 chromosomes, 2 copies of each chromosome, which is why there's X's, and 2 homologous pairs. These 2 chromosomes, these 2 duplicated sister chromatids separated and went into this daughter cell that's haploid. We then will separate these sister chromatids from each other during meiosis 2, which will make daughter cells that are haploid, but rather than having 1 copy of each, they will have, or 2 copies, they'll have 1 copy. And the same thing is true 4 over here. The same process occurs for these two homologous chromosomes. I hope this helps with just some basic overview of understanding how we create gametes and genetic diversity amongst species, and we will be continuing this work after break. Have a great day.